Hello, this is chapter 21, deals with skin and eye infections. We are now entering into the section in this textbook where each chapter is going to look at different of the body systems and the various types of microbial infections that occur within that particular system. Obviously, there's going to be some overlap between some of the systems. And so sometimes there kind of has to be sort of a judgment call as to which this disease affects multiple systems, so where are we going to put it? And so the other thing that you'll find with each of the remaining chapters as we look at the different systems, such as this one, the integumentary system or the skin, and then also including eye, because eye has a lot of infections, it's going to be broken down just kind of a very basic general anatomy of that particular system. I won't go to a lot of detail. Your book does on some of it, but this is not an anatomy class. So that material should be learned in anatomy class. I'll go over just some general basics of it. And then the way it's divided, it separates it into like bacterial infections and then viral infections and fungal infections and then protozoan infections and helminths infections. Some of the systems aren't going to have all of those different groups of microorganisms, but that's the way it will be divided into those sections. So hopefully that makes sense to you. So with this, the integumentary system, which is the skin, the skin is the largest organ in the body. It is, in basic skin anatomy, it's divided into different layers. You have the epidermis, the dermis, and the subcutaneous layer. The epidermis is the outermost layer. What you see on the surface is dead cells. Sorry, I hate to disappoint you, but it's all dead. The cells underneath are constantly going through mitosis, producing new cells and pushing the previous made cells towards the surface. The epidermis does not have a blood supply to it, and so that's why the cells end up dying. Nutrients can only diffuse so far, and so by the time it reaches the surface, actually before it reaches the surface, the cells die. And then those dead cells are constantly being sloughed off. As we discussed in the section on the immune system, the first line of defense, that sloughing off of those dead cells, can be very beneficial and helpful in helping to prevent infections. Keratin is a protein. It gives tough and waterproof properties to the skin. Your sebaceous glands are associated with hair follicles. They do secrete sebum. We mentioned this also with the first line of defense. You also have sweat glands that are present in the epidermis. Sweat contains salts. A lot of microorganisms cannot tolerate high salts. It contains the enzyme lysozyme, which, as you've seen previously, is microbicidal. It will kill things like bacteria. And there are some other peptides that are present that have antimicrobial properties to them. And so this is a picture. If you've had A&P, hopefully this kind of looks familiar. On the far left, we have an actual micrograph or picture of the epidermis and the dermal layer that is below it. And then on the right, we have the schematic drawing of these different layers. So the upper layer up here is the epidermis, and then you have the dermal layer beneath it. The dermal layer does have blood vessels, more down here towards the bottom. From a microbiology standpoint, we're always concerned about that with infection. You do not want, we talked about this with the immune system, you do not want any invader, whether it's a bacteria, virus, whatever it is, to get into the blood system. Because once it's in the blood system, that's going to make it so much harder then to treat. Some medical terms associated with the skin. Sometimes you hear the term an abscess. That's basically a localized collection of pus. Pus is dead cells, dead tissue debris, etc. Bulla is a fluid-filled blister that's less than 5 millimeters in diameter. If it's a deep pus-filled abscess, it's carbuncle. Crust is dried fluids on the skin surface. It's usually covering a lesion. A cyst is a sac It's an encapsulated sac. It's filled with fluid. It can be semi-solid or it can be gas. Folliculitis is inflammation around the hair follicle. It's just like, it looks like a localized little rash. Anything that ends with itis means inflammation. A boil or furuncle is a pus-filled abscess. It's due to the infection of the hair follicle. And the macules are spots on the skin that are discolored. So with various diseases, you're going to see some of these terms. And no, we're not done yet. Papules are small raised bumps on the skin. A pseudocyst is a lesion. It kind of looks like a cyst, but it doesn't have a nice defined boundary like a cyst would. Purulent means it's pus producing. Pustules are fluid or pus-filled bumps on the skin. Pyoderma is any pus-producing infection on the skin. An ulcer is an open sore. Vesicle is a small fluid-filled lesion, and a wheal means that it's usually swollen, little inflamed area on the skin that's itchy. It may have a burning sensation to it. So we use these different terms to distinguish between different, not really side effects, but different things you see with skin infections, the different symptoms, the different visual things that you may see. And so this is a picture showing acne on the skin here, and this is a schematic drawing of what it is. You've got pus right underneath the skin. Acne usually is associated with folliculitis, or the inflammation of a hair follicle, and then the whitehead that's near the center of the cheek over here on this picture, you can see it is an infected hair follicle, and it's producing that pus, which is, like I say, dead cells, dead tissue in there. So this is a picture of these different types of skin lesions that I mentioned just a bit ago. Sometimes it's a little bit easier to see. A cyst, remember, it is encased in a sac in here, and so if you were to do a procedure to remove that cyst, you do want to remove the sac in its entirety in there. So you're just making distinctions between is it an open lesion like an ulcer, or is it a small bump? What does it look like? Different diseases are going to have different manifestations of it, so that's why it's important to know the difference between them. Simple review of the eye anatomy. The lacrimal apparatus. This involves the lacrimal gland that actually produces the tears, and then which is above the eye, on the outer portion above the eye, and then every time you blink, the tears that are being produced, they're going to flush across or move across the surface of the eye. It helps to lubricate the eye. It helps to prevent infection. You do not want anything to enter through the pupil and go deeper into the eye. And then it will drain through the tear duct, which is right near the nose. It's going to drain, and then into part of the lacrimal apparatus that's right on either side of the nose. That's one of the reasons why if somebody is crying, oftentimes people think, oh, the tears are produced near the tear ducts next to the nose. No, it's just you're producing more tears faster than what it can drain out. It's also why if you get hit on the nose, oftentimes your eyes are going to water or tear up because it's pushing that excess tears out from where it drains in next to the nose. The conjunctiva is a membrane that's on the surface of the eye, and this is, because it's on the surface, it's a common location for infections to occur. So in this picture, it is showing here is the lacrimal gland up here. As I said, it's above the eye. It's going to produce the tears, which now will flush across the eye. They are going to drain into the tear ducts and then be stored into lacrimal sac that stores the tears, which eventually drains down into the nose. Which also explains why if somebody's crying a lot, they tend to have a runny nose, because that's where the tears that drain through the tear duct are going to end up. When you look at the eye itself, the conjunctiva is this outermost membrane covering the eye. Because it's the outermost one, that's why it can become infected so easily. And like I said, the pupil in the middle, that's that black hole in the middle of the eye when you're looking at someone's eye, that is a hole that allows light to come in that normally comes back and hits on the back of the eye so you can see whatever image. You don't want something to come in there because once it's in the eye, it could travel here. This is the optic nerve. It can leave through the optic nerve. It now has access to the brain. It's in the brain. So that's why you want to be very careful and try to prevent infections through the eye. So with eye infections, conjunctivitis is inflammation of the conjunctiva. Blepharitis is inflammation of the eyelids. Inflammation of the cornea is keratitis. And dacryocystitis is inflammation of the lacrimal sac, where those tears are being produced. So conjunctiva, the inflammation of it, here you can see this extremely, on the far left, this extremely red eye. As we'll see in a bit, pink eye is an example of an infection or an inflammation of the conjunctiva. Infection of the eyelids is in this middle section here, and then inflammation of the cornea is in the far right. So now this is where we're going to start and look at specific organisms. As I said, we're going to start with bacterial infections first. Sometimes this is divided according to organisms. Sometimes it's divided according to the disease. If it's only one organism that causes that particular disease, then I tend to put that organism up at the top here. If there are several organisms that are causative agents, I tend to put the disease, and then underneath I will list the various organisms. So in this case, we're looking at bacterial infections. We're looking at Staphylococcus. In this genus, and there are several species, overall, Staphylococcus is a gram-positive bacteria. It is classically found in grape-like clusters. That's how it's usually described as. Just so you know, the grape-like clusters are more easily to be seen when Staphylococcus is grown in a liquid culture versus a solid. But still in solid, you may often see it. One thing about Staphylococcus is that it can tolerate high salt concentrations. Most bacteria cannot. And so for this reason, Staphylococcus can grow in a media called mannitol salts agar. This is a media that is made to have a little bit higher salt concentration. So it's a selective media in that typically only Staphylococcus will grow on it. Staphylococcus is commonly found on the skin. You can probably test anyone's skin. You can probably test doorknobs and tabletops. It's pretty much everywhere. So it's commonly found on the skin. In more virulent strains of it or more infectious strains of it can be contagious. There is a problem with anti-resistant strains. Some of you may have heard of MRSA, which is a methicillin-resistant Staphylococcus aureus strain. That is a problem. So you might have to be careful and run some additional tests to see if you're working with an antibiotic-resistant strain, because that might make a difference as to what antibiotic you would prescribe to your patient. Something to keep in mind is that Staphylococcus does produce an enzyme that is called beta-lactamase. This is important because beta-lactamase is essentially going to break down penicillin. Penicillin normally you think of as being an effective antibiotic against gram-positive bacteria. So your natural first thought is going to be, oh, Staphylococcus is gram-positive, so we can use penicillin. And no, you can't. You're going to have to use a different antibiotic because Staphylococcus has developed so that it produces this enzyme that will break down the penicillin, so it's worthless against it. It can produce several different enzymes to help it penetrate once it gets past that skin barrier. If it can, oftentimes Staphylococcus will be on the surface, but it can't penetrate. But if you get a cut, you get a scrape, some type of an abrasion, That's like opening the door. Okay, now it's in. Now it produces enzymes that allow it to penetrate deeper into there. With this section, just so you know, because I know your book goes into a lot of detail with a lot of the various diseases in this chapter and in the following chapters. What I'm going to be focusing more on is what organism causes what disease. So organism A causes disease A. Organism B causes disease B. With some of these, there are going to be some unique things that you need to know. Do you have to know all the details? No. In this case, like Staphylococcus, because I made a point of talking about the beta-lactamase and that you cannot use penicillin in, that is something unique to Staphylococcus. So that is something I would want you to remember with this. In this picture on the left, it is showing a mannitol salts agar plate. You'll work with this in the lab. The agar is normally this pinkish color, as you can see on the right side of the plate. But notice the left side's yellow. That is because mannitol salts, number one, it's a selective media, as I said previously, that only Staphylococcus will grow on it because of the high salt concentration. It's also a differential media. Remember when I talked in one of the previous chapters about media and how to grow microorganisms? Differential media, you've added something to it so you can usually very easily just look for a color change. And then you can distinguish then between two different bacteria on here. Staphylococcus aureus is on the left. Staphylococcus epidermidis is on the right in the pinkish area. Staphylococcus aureus is able to ferment mannitol, and it produces acid. And because it's producing acid, it has turned, there's a pH indicator in here, and it has turned this left side yellow. Staphylococcus epidermidis, it can grow in here, but it doesn't ferment and produce acid. So therefore, it did not change the pH, so it stayed the pink color. So you can just look at it. You see growth on a mannitol salts agar plate, you know it's Staphylococcus. You see that it's turned it yellow, you know that it's Staphylococcus aureus. Over here on the right is an electron micrograph, specifically a scanning one, that is showing how you have these grape-like clusters. It's cocci in shape. That's classic Staphylococcus. So what are some of the bacterial infections? Number one, Staphylococcus aureus, which of the various strains of Staphylococcus, this would be the most virulent one, meaning the one that causes the most diseases. Pyoderma is skin infections, purulent. You can treat it with antibiotics. You can prevent by using good hygiene practices. It can also cause scalded skin syndrome. This you can treat with antibiotics and make sure you are supplying fluid so the individual does not get dehydrated. Scalded skin syndrome, there's a picture of it in a moment, and it's going to look kind of like a horrible sunburn and you're peeling afterwards. So some of you may not have seen that before. Impetigo, some of you may be familiar with this. It looks like encrusted sores or lesions, and you can treat this with antibiotics as well. Do you have to know which antibiotics? No. In this case, you know it's not going to be penicillin. And so this is showing some skin infections caused by Staphylococcus aureus, where you have these boils, these pus-filled lesions. This is a newborn that has a staphylococcal infection. They have scalded skin syndrome. So like I was saying, it looks like, as I said, a horrible peeling after a sunburn. You get this peeling of the skin along here. Once it has run its course, usually there's no scarring. It'll completely heal from that. It's just one of those things where it looks horrible while the person has it, but they will, in most cases, will completely heal from it. The one thing I should also caution you as we go through this chapter and the remaining chapters where we're talking about diseases and they will have pictures, they tend to sometimes pick some of the worst of the worst pictures. So I'm just preparing you for that. You're forewarned. Here is an individual that has impetigo in this area right here. You have a lot. One of the things about it typically is encrusted on the sores there, the lesions. Streptococcus. So as we're moving on to another genus. Streptococcus is gram-positive. It's usually in chains. So Staphylococcus is usually in clusters, but Streptococcus is usually in chains. Streptococcus is classified as facultative anaerobes, meaning it can grow in oxygen and in a regular lab setting. It can grow. It's going to look real teeny tiny colonies on a petri dish. It's not going to be happy. When we grow it in the lab, we usually put it in an anaerobic, like a candle jar. It prefers to have a lower concentration of oxygen for growth. There are different strains of Streptococcus, and they're first usually grouped as non-hemolytic, alpha hemolytic, or beta hemolytic. They can produce several different enzymes, just like Staphylococcus can. The non-hemolytic, if you grow the Streptococcus on a blood plate, you're not going to have any hemolysis or any lysis of those red blood cells. They remain fully intact. Alpha hemolytic, when it grows, it's going to become like a greenish color to it. Beta hemolytic strains will completely lyse the red blood cells to the point where you can have a blood plate which is going to look red and it's opaque. You inoculate it with a beta hemolytic streptococcal strain, it is going to completely clear that plate. Where I've seen where you could read something through the plate. That's how much it will lyse those red blood cells. Those are not the ones you want to see because those are usually very virulent, very definitely a pathogenic strain. Streptococcus pyogenes is one of the species that is responsible for several different diseases. In this picture, you can definitely see that it's cocci, that it's in these long chains. If you look at something like this, can you identify a bacteria solely by its gram stain? No, you cannot. But it can certainly give you some clues. Looking at this and seeing the chains, my immediate thing is that's not Staphylococcus, that's for sure. Chains, cocci, gram-positive, I'm going to be leaning towards Streptococcus. So what is Streptococcus pyogenes that you just saw on the previous slide? What does it cause? It can cause cellulitis, which is an infection in the dermis or the hypodermis, so not on the outermost layer, but deeper than that. You can treat that with antibiotics. You can use penicillin with streptococcal infections. You can have erysipelas, which is an inflamed patches on the skin. You can treat that with antibiotics. Erythema nodosum, little red nodules on the skin. You can treat that with anti-inflammatory drugs. Impetigo, those encrusted sores or lesions, you can treat that with antibiotics. Another thing that Streptococcus pyogenes can cause is necrotizing fasciitis. You can treat this with antibiotics, but this is a very serious condition of where the bacteria has been able to invade the body, and it's known as the flesh-eating bacterial syndrome. It starts destroying the tissue. You need to start treatment right away on this. You'll notice as we go through this, and I go through the different diseases that are caused by particular bacteria. I will tell you whether you can treat it with antibiotics or not. Later on, there may be sometimes where we'll say there is no treatment. Do you have to know specifically which antibiotic? No, you do not. Your book tells you that, but you don't need to know that for this particular class. And so in this picture, under A on the far left, it is showing cellulitis. And then you can see various stages of other rashes. All of these are caused by Streptococcus pyogenes. You'll notice, remember that Staphylococcus also caused rashes, so sometimes it can be a little difficult to figure out which one is it. This is when something becomes necrotic, it means it's dying, it's being destroyed, and that is what is happening in this picture on the far left on this patient's leg. Now, oftentimes what you have to do, and it's going to be painful, is go through, and it's called debriding. You have to go through and remove any of the dead tissue, because once it's dead, it's dead. You need to remove that to try to help remove the infection. That's part of the treatment. Is it painful? Yes. Is it something that has to be done? Yes. So with this necrotic fasciitis, not only does Streptococcus cause it, but also there are other bacteria that can cause it. Klebsiella, which is a gram-negative bacteria, it can cause it. Clostridium, which is a gram-positive, can cause it. Escherichia coli, which is commonly referred to as E. coli, which is gram-negative. Staphylococcus aureus, that we just talked about previously, is, as you know, gram-positive. And Aeromonas hydrophilia, which is gram-negative. All of these can cause it. So this is where, as you can see, some are gram-positive, some are gram-negative. If your patient is diagnosed with this disease, you need to figure out what the causative agent is. You need to figure out, is it a gram-positive or a gram-negative? Because that right there is going to make a difference as to what your choices are for what antibiotic you're going to use to treat it. Pseudomonas aeruginosa. This is a gram-negative bacteria. It's bacillus shaped, so it's rod shaped. This is an opportunistic organism. It's very commonly found in soil and water. It's one of those ones that in most healthy people, it's not going to cause a problem. However, if you already have an infection, if you have a cut where it's like, you know, if you open the door, it's going to take advantage of that. It's aerobic, so it likes oxygen to grow. One unique thing about Pseudomonas is that, especially aeruginosa, is that under certain conditions, it produces a pigment and it fluoresces under UV light. Sometimes it fluoresces. It depends on which pigment it produces, and that's going to be dependent upon the nutrients available, the environmental conditions. Sometimes it kind of fluoresces a greenish yellow color, and then we currently have a strain of Pseudomonas aeruginosa in our lab, teaching lab, and it will often fluoresce kind of a blue color. Pseudomonas aeruginosa can produce several different virulence factors that helps it, once you've opened the door, to invade deeper. One problem with Pseudomonas is, even though it's opportunistic, once it's in, one of the problems we have with it is that it's becoming resistant to most antibiotics. Pseudomonas kind of grows on everything, and so it's hard to control. It's hard to get rid of it. So what are some of the bacterial infections caused by Pseudomonas aeruginosa? There's a condition known as hot tub rash, associated with spending time in a hot tub where the water gets contaminated. This can be treated with antibiotics. Otis externa, which is commonly referred to as swimmer's ear, also treated with antibiotics. Various wound and burn infections are notorious for getting infected with Pseudomonas aeruginosa. Once again, you can use antibiotics. And then keratitis, which, as we mentioned earlier, is inflammation of the cornea. Pseudomonas, you can try to treat it with antibiotics. Just be careful because of the drug resistance that seems to be increasing with it. This is the hot tub rash over here on the left, and then swimmer's ear or Otis externa on the right. You can see that there in here there's definitely an infection going on. Pseudomonas is shown here on a plate growing. Excuse me. The individual colonies on the far left here. In the middle, you can kind of see that it's producing a pigment that you can see just under just normal regular light in the middle. And then if you put it under UV light, you get the glowing like this. It's kind of cool to see, as long as it's not on you. Propionibacteria acnes is a gram-positive bacillus. It is aerotolerant, so it can tolerate oxygen at lower concentrations. It's typically found on the skin. It can be the causative agent of acne, where acne is where with the hair follicles, and you have the sebaceous gland secreting the sebum, and it gets infected. And what happens is the hair follicle ends up getting clogged along there. Treatment for it is going to depend on the severity of it. If it's mild, you just sort of let it run its case. In real severe cases, you may give salicylic acid and laser treatment. You can use antibiotics to treat. In some cases, preventative, just use good hygiene practices. And unfortunately, a lot of it has to do with, puberty and increased hormones and increased sebum production, etc. So over here on the left in this schematic drawing, it is showing how you have right here a normal the skin pore, the hair follicle coming up right here, and here is your sebaceous gland right here that's producing that sebum. With a whitehead, what has happened here is you have a lot of buildup of the lipids, and it has become infected. A blackhead is when it becomes oxidized. And then over here, you can see there are several cases throughout along the nose. Bacillus anthracis. This is a gram-positive bacteria. It produces endospores, which is going to be a problem. It's a facultative anaerobe. As the name implies, it is the causative agent of anthrax. So there's different types of anthrax. Cutaneous anthrax is where it's going to enter through the skin, and it forms a nodule, and you get this massive dead skin tissue on the skin. You can treat this with antibiotics. There is a vaccine that is available. It is not for the general public. It is reserved for certain select groups. Military is usually vaccinated against this. And the reason for that is because there is concern of Bacillus anthracis being used as a biological weapon in bioterrorism. It commonly grows in the soil. It's fairly easy to grow, but if you become infected with it, if it's cutaneous anthrax, you can treat it with antibiotics. This is shown here, an example of cutaneous anthrax, and this is shown with it growing on blood agar plate. When I was in school way back, we actually worked with Bacillus anthracis. As we moved through the different systems, like respiratory, there's another form of it, which is very, very deadly. We worked with Bacillus anthracis as an undergrad, which I look back now and I'm kind of like, I can't believe they let us do that. Just since 9/11 and concerned about bioterrorism, only certain labs are allowed to work with Bacillus anthracis for obvious security reasons now. If I were to request a culture, try to order a culture of it, I would be getting a visit from some government officials. It is very tightly regulated now, like I say, for security reasons. Still continuing with bacterial infections. Conjunctivitis, which is pink eye, is highly, highly contagious. Oh my goodness, they have outbreaks of this sometimes, like in daycare centers, because kids touch everything, including their eyes, and then they continue to touch each other and all the toys, and it just goes like wildfire through it. So it's highly, highly contagious, regardless of what the causative agent is. There are several different things that can cause it. Haemophilus influenza, which is a gram-negative bacteria. Moraxella catarrhalis is a gram-negative. And then you've got Streptococcus pneumoniae, which is gram-positive, and Staphylococcus aureus, which is also gram-positive. All of these you can treat with antibiotics. Because it is highly contagious, you want to, number one, get a handle on it as quickly as possible, and don't be touching your eye. It may be itchy, you may want to, but don't touch your eye. This is a picture showing someone with pink eye. It gets its name, if you've never seen it before, because of how red the eye looks. And you do get a lot of this pus being formed, which is very aggravating. This discharge, and then it can dry and become crusty, and usually it's treated by antibiotics, usually either eye drops or a lotion that you, like a cream that you put right along, typically along the bottom eyelid. Neisseria gonorrhea. This is a gram-negative coccus. With the gram-negative bacteria, there are very few of them that are pathogenic that are cocci. This can cause an infection with the eye. One is ophthalmia neonatorium, meaning it's an eye infection on newborns, and also inclusion conjunctivitis. The way you treat this, because it involves newborns, is you give the antibiotic immediately after birth. You need to treat it immediately so you do not have long-lasting results from it. And so you can see that what happens here is you have a purulent, like a pussy discharge, and then it tends to kind of crust over. So you want to clean this off. You want to give the ointment. If it is left untreated, it can result in blindness. So that's why they do it immediately after birth. Chlamydia trachomatis is gram-negative, it's anaerobic, it's an obligate intracellular parasite, so that means it has to grow inside your cells. It can also cause inclusion conjunctivitis, which can be treated with antibiotics. It can also cause what is known as trachoma, also known as granular conjunctivitis, which can also be treated with antibiotics. Staphylococcus epidermidis can cause keratitis. Remember, that's inflammation of the cornea. This can also be treated with antibiotics. So at least so far, the diseases we've been talking about can be treated with antibiotics. So that's a good thing. What happens with trachoma? The eyelashes can start turning inward, and then if left untreated, that can be a problem for your vision, obviously. If we move on now remember, all of this is dealing with the skin and the eyes. So those were all the bacterial infections. Now we're going to move on to the viral infections. Viruses, if you remember, their classification is a little bit different than everyone else. We do not have genus and species with viruses. It's just named down to family, and that's it. So I will be giving you the family name. I will tell you usually at the very beginning whether it is DNA or RNA, and whether it's single-stranded or double-stranded. So DS means double-stranded, or SS is single-stranded. So with the viral infections, here's the human papillomavirus, HPV. It is a double-stranded DNA virus. It can cause papillomas, which are a fancy name for warts. It's transmitted by direct contact. There are different types of warts that it can cause. Common warts are typically found on the fingers, the nails, your hands, so on those extremities. Plantar warts specifically are found on the foot. Flat warts are those that can be found anywhere on the body. They're typically very small and they're smooth. They're kind of flat nature to them. Filiform warts, these tend to be very long, kind of thread-like. They grow typically very quickly. And then also genital warts. Now, genital warts will be discussed later in a different chapter. Treatment for the warts, there's usually multiple options that you can use. Some people use liquid nitrogen that basically freezes them off. Some people use laser treatment. You can burn them, you can cut them. So there's several different methods. That's for you to discuss with the patient to discuss with the doctor as to what form of treatment is going to work best for them in that situation, in the location of the wart, etc. So here is an individual on the far left here under A. Is a picture of a toe that has several of the plantar warts on it. And then a filiform wart right here is seen. Like I said, those tend to grow very quickly. Herpes simplex virus 1, which is referred to as HSV1. You'll find oftentimes with the viruses we use abbreviations with them. This is also known as oral herpes. It's a double-stranded DNA. It is highly contagious. Now, some individuals can be asymptomatic. They can still be contagious, but they are not showing any symptoms. Some people, the virus goes latent, and it can remain latent for a long period of time. The recurrence of it, we don't know exactly what triggers it. It seems to be stress and or environmental factors as to what triggers it to come out of this latent or dormancy state. Usually for the HSV1 virus and oral herpes is typically on the lips. It's often referred to commonly as cold sores or fever blisters. In order to treat, you try to just reduce the symptoms of it. Cold sores and fever blisters are different than canker sores. Just put that out. If it's found on the fingers, it's known as whitlow. They just give it a different name. Sometimes wrestlers or athletes will have an outbreak on their neck or their shoulders or the trunk of the body. Is known as herpes gladiatorium. And keratitis, which is once again inflammation of the cornea with the eye, that can be treated with antibiotics. So here's an example of a cold sore that's being caused by HSV1. Human herpes virus 6, HHV6, and human herpes virus 7, HHV7. Both of these are double-stranded DNA. They can cause roseola, which is, there's a couple different names for it. As you can see, it's a rash. It's fairly common in children. It can be spread by direct contact or aerosol, sneezing, things like that. Normally, no treatment is necessary for it. Parvovirus B19 is another viral infection. This is a single-stranded DNA. This also is known as a causative agent of fifth disease or erythema infectiosum. This is highly contagious. It has a rash. It's more common in children. Once again, oftentimes no treatment is needed. And so you can barely see here the rash, the roseola. This is a mild infection. And then fifth disease, also a rash. You can see on the cheek here, it's more pronounced than elsewhere on the body. You're starting to get an idea that a lot of these cause rashes, but it can be kind of hard just by observing the rash to figure out what the causative agent is, which is why it's necessary to run your lab tests to definitively identify what the causative agent is. And then that way you can improve what your treatment's going to be. Adenovirus is a double-stranded DNA, can cause conjunctivitis. You tend to get more of a watery discharge than the thick, gooey discharge you got with a bacterial infection. So that's one way that can kind of help you distinguish between them. It is highly contagious. There is no treatment available for this. Remember, viruses, it's harder to treat than bacterial infections. So now we're moving on to fungal infections. Mycoses. Mycology is a study of fungi, so mycoses is a fungal infection. Cutaneous mycoses are going to be superficial, just on the surface of the skin, the hair, and possibly the nails. Subcutaneous, now the infection is deeper. It's gone past the epidermis and it's in the dermal layer. Systematic mycosis, it's spread throughout the body. That's definitely going to be a problem. So when we look at the fungal infections, first one we look at is caused by Tinnitus. Where is the disease Tinnitus? It's caused by certain molds that are found in the soil that are referred to as stromatophytes. The species, trichlorophyton, epidermophyton, and microsporidium. The general name of this is referred to, of Tinnitus, is ringworm. You can treat this with antibiotics that are basically antifungal drugs. Common names for the Tinnitus or the ringworm and where they're located in the body. Tinea corporis is known as ringworm. It's throughout the body. Tinea capitatis is ringworm that's found on the scalp. Tinea pedis is known commonly as athlete's foot. Obviously, it's found on the feet. Tinea barbae is a barber's itch. It's around, if somebody has a beard, it's typically around that area. Tinea cruris is jock itch in the groin area. And Tinea unguineum is the toenails or the fingernails. So it can be found, obviously, different species, different areas, different names of them. And so here's the barber's itch around the area of where, if you had a beard, where it would be. Athlete's foot, obviously on the foot area here. And then this is a close-up picture of ringworm. That's right in here. It has a misnomer of being called ringworm. It's not a worm, it's actually a fungus. To diagnose it, you have to grow it on an agar, a Sabarod agar. Dextrose agar is specifically designed for, or selected for fungi. The pH is dropped a little bit, so bacteria don't like it. This is showing on a plate the fungus as it's growing on it. Aspergillus fumigatus and Aspergillus flavus. They are causative agents of aspergillosis, which usually starts in the respiratory system, but then it can spread. It's an opportunistic, so in most cases, most people it won't bother, but once again, like I say, you open the door and allow it in, allow it to spread, then you can have a problem. This is often transmitted as a nosocomial infection, meaning it's acquired in a healthcare facility, a hospital, maybe a nursing home, etc. Especially with patients who are immunocompromised. They're already stressing out their immune system. It's already not working at 100% because it's trying to deal with some other infection or some other situation. And so it just makes it easier for the secondary infection to come in. You can treat this with antibiotics. So here is someone, you can see the lesion here, with who's suffering from cutaneous aspergillosis. And this is what it looks like under the microscope. This is what you call the kinetophore, and these are all these little dots in here are spores. Candida albicans causes candidiasis, rash on the skin. This can also, besides the rash on the skin, cause yellowing of the nails. It can cause hardening of the nails. It can also cause a rash, typically seen in skin folds, called intertrigo. And then in both of these cases, your immunocompromised individuals are going to be at a higher risk for contracting these. You can treat it, once again, with antibiotics. So this is the rash that's being caused by the Candida albicans. And then you can see here on this nail that it looks a little bit more brittle. It looks yellowish in color. Obviously, it doesn't look healthy. That is because of the fungal infection. And then once again, this is what it looks like when it's grown on the petri dish in Sabouraud dextrose agar. You'll notice that when fungi grow on a petri dish, they're larger, usually much larger than what bacteria. And yes, they sometimes have a crusty or a fuzzy look to them. The surface is definitely different than bacterial. Sporothrix sketchikai causes sporotrichiosis, also known as rose gardener's disease or rose thorn disease. It causes a rash on your arms that looks like you had a fight with the rose bush and the rose bush won. So it will start as a small little ulcer where, like I say, it looks like a thorn prick, and then it spreads to the lymphatic system. You can treat it with antibiotics. Moving on to protozoan infections. Acanthamoeba is a free-living amoeba, protozoan, commonly found in soil and freshwater. It can cause keratitis. Usually it's going to be mild, but if you do not treat it, it can advance to a very severe infection. So treat things as soon as you realize you have something. Immunocompromised individuals are higher risk. If you wear contacts, you're going to be at higher risk, because if you have contacts and you get an infection, the contacts are kind of helping to hold it in there. So just know it's going to put you at higher risk. Treat it with antibiotics. So this is just showing how some of these parasites that are very common in water that's not chlorinated, so in a pond, a lake, a river, whatever, that the cyst, protozoans often form a cyst, and that's what enters your body. Either you swallowed some water, it got in your eyes, etc., and that's how it enters. So this is what the cyst looks like. Protozoans usually have multiple forms throughout their life cycle. We don't get into it because this is general biology. So usually what enters the body is the cyst form, and then it converts over to a different form. And oftentimes use a fluorescent dye to try to look to see if there's any damage, and that's what's been done here on this eye, where they've added a fluorescein application, and then they shine the highlight on it to see the damage that has been done. Helminths. Helminths are worms, different types of worms. The loa loa is a nematode. It's also known as African eyeworm. It causes loesis. Deer flies are going to be the vector. One thing you will find with protozoans and helminths is that often there is a vector. There is like an intermediate, it may be a biological vector, like a fly, a tick, a mosquito, or it could be a mechanical vector, something inanimate that is going to transfer from one place to another. So in this particular case, deer flies are the vector. Adult worms can survive in the subcutaneous tissue for years. That may not be something you want to hear, but it can. There's usually, it infects the eye. There's usually no lasting damage to the eye as the worm moves across the conjunctiva. You might see, the individual who's infected might see something kind of squirming across. You can treat it by surgically removing the worm from the eye or from the skin. Sometimes with worms you have to be careful to make sure you remove the entire worm. So you usually have to get antibiotic treatment in addition to trying to remove it. So this shows, like I say, they're going to give you the extreme cases here where someone became infected and the worm was removed. In this case, they said the worm that was extracted from the conjunctiva was 55 millimeters long. So, 55 millimeters, that would be 5.5 centimeters. And I'm trying to do math real quick here. That's over 2 inches. That's long in your eye. I know when I lived in New Mexico, I don't remember hearing what kind of a worm it was. It may have been like a low worm, where there was an individual that came into the emergency room and was complaining that something was in her eye. She could see something was in her eye, and nobody was believing her. They thought she was crazy until finally somebody looked at her and they thought, wait a minute, I thought I did see something, and they removed a worm from her eye. Just things, to give you nightmares. So these are obviously not all the diseases, but we go through some of them. Are you going to see all of these in the course of your career? I hope not, but you may. You never know where you're going to be working, and so you may see some of these. So like I said, this is the first chapter where we're looking at a particular system. From now on out, we're going to go through all the various systems. Some of them are combined together, but we're going to go through them, and it'll be set up in the same manner as dividing it by bacterial, viral, fungal, protozoan, helminths. What's the causative agent? And as you go through this, as I said at the beginning, I want you to focus not so much as to how it's transmitted and what makes it virulent, but more of this organism causes this disease. As an example, like if I were to set up a matching section, would you be able to answer that, fill that out? Just so you know, I always try to balance. Yes, you need to know some of this information, but it is an introductory course. Yes, it is technically a sophomore level course, obviously a college level course. I know it's a lot of material, but as you're studying this from this point on, be looking at it, whether you make a table, whatever works for you, index cards, whatever. But like I said, just put this organism causes this disease.